The experiment on chemiosmosis carried out by Jagendorf in the 1960s proved direct evidence for the chemiosmotic theory. Jagendorf used isolated chloroplast thylakoids and manipulated the pH to create a proton gradient artificially. So the thylakoids were first placed in an acidic solution to saturate them with protons. They were then rapidly transferred into an alkali solution, creating a proton gradient across the membrane. And it's also worth noting that the experiment was carried out in the dark, so the proton gradient served as a source of potential energy, mimicking the conditions created by the light-driven electron transport chain in photosynthesis. So the thylakoids were first placed into an acidic solution to saturate them with protons. So if the external conditions are highly acidic, the high concentration of external hydrogen ions could theoretically alter the gradient. So this causes a higher amount of hydrogen ions to diffuse into the thylakoid lumen. So we actually have a look at what's happening. So outside the thylakoid, there's low pH causing a high concentration of high hydrogen ions. So hydrogen ions diffuse down a concentration gradient through hydrogen ion channels into the thylakoid lumen. So there's no light, so photosystems are not involved. Thylakoids were then rapidly transferred to an alkali solution, creating a proton concentration gradient across the thylakoid membrane. So the solution also contained a high concentration of inorganic phosphate and ADP. So the movement of protons through the membrane catalyzed the generation of ATP by ATP synthase. So inside the thylakoid now has a high concentration of hydrogen ions. Outside the thylakoid, there's now a low concentration of hydrogen ions. So therefore, the protons diffuse through ATP synthase, which produces ATP from the ADP and PI. So it wants to see how the ATP was detected. So it used a method that used the enzyme leucophase, which catalyzes a reaction with leucoferrin. So in the presence of ATP, it produces light. So the amount of light emitted is directly proportional to the concentration of ATP present. So Jagendorf could measure this light using a photometer or a luminometer to confirm ATP synthesis. So the ATP was synthesized in the absence of light, demonstrating this proton gradient alone was sufficient to drive ATP formation. So it's support for the chemiosmotic theory. So the results directly confirm that ATP synthase depends upon a proton movement force, so a gradient of protons across the membrane, and not the energy from light or external factors directly.